Hi, welcome to the course Life Science for Educators. This is part of the B.Ed Honours programs, and this is a video to help guide you through the presentation or the preparation of the seminar task. This video deals with seminar task 2, the endomembrane system. In seminar task 2, you are going to be discussing the endomembrane system. You have to describe the endomembrane system and its significance to the cell. Now, I've included a diagram here from the nucleus showing the endoplasmic reticulum and the Golgi apparatus budding off vesicles, which then merge with the cell membrane. The information to make proteins is in the nucleus. That is transcribed, it's translated on the ribosomes. The proteins are then absorbed or taken into the endomembrane system. As you can see, that little green molecule. Then from the vesicles, it is maybe some kind of carbohydrate is added, and it's then packaged in the Golgi apparatus, and then it is merged with the cell membrane. So consider the significance of that entire process and how the endomembrane system works together from the making of the proteins to the fusion with the cell membrane. Okay, so you'll be using the biology textbook Campbell, the 10th edition. I have put up a copy on Ulwazi, but please make sure you use the textbook as your main source of information. Build your presentation from the textbook. Check the topic against the concepts in the textbook and ensure you cover all the aspects. So you'll be working with chapter 6. It's pretty much the same content as in seminar task 1. However, your approach is very different. You're describing the endomembrane system and its significance to the cell. So I'd like to briefly take you through a tour of the plant and animal cell using micrographs. Okay, this tour of the cell is from the book The Cell, a workbook for biology students. It was produced by Sackett Trust, and the author was Liz Dilly, who has made many of the focus textbooks. Okay, this is a micrograph of a rat liver cell. There you saw the plasma lemma. There's the nucleus. Please notice the mitochondria. And you can see the endoplasmic reticulum there around the cell. This is a plant cell from a clover. You can see there is a chloroplast, but note the plasma membrane is on the inside of the cell wall. There is the nucleus. The endoplasmic reticulum, there are parts on either side of the nucleus, and there is the vacuole and the mitochondria. So these are micrographs of both a plant and an animal cell. Now, this is an transmission electron micrograph of a cross-section through the nucleus. You can clearly see the nuclear pores, the nucleolus, the nuclear envelope, which is a double membrane, and the chromatin network. There you can see the nuclear pores quite clearly, and you can see the endoplasmic reticulum surrounding the nucleus. Now, this is a scanning electron micrograph showing a 3D version. There you can see the nucleus with the nuclear pores and the endoplasmic reticulum around the nucleus. So I'm showing you these so that you can see what they actually look like if we take electrographs. Now, here is the endoplasmic reticulum. The bottom shows smooth endoplasmic reticulum, so there are no ribosomes attached. This is now showing you rough endoplasmic reticulum, and those black dots are the ribosomes where proteins are synthesized. So that's the difference between smooth endoplasmic reticulum and rough endoplasmic reticulum. There is a scanning electron micrograph showing the endomembrane system, and there you can see the nucleus. And so you can see the endomembrane system surrounds the nucleus. There is a micrograph of the Golgi apparatus. That was in an animal cell. There it is in the plant cell, and you can see the stacks and the vesicles coming off. There is another picture of the Golgi apparatus, and I'm also showing you here the lysosomes. The lysosomes contain digestive enzymes, so it's very important that they are enclosed in that membrane. So to end this input session, I'd just like you to watch this video on the endomembrane system.
The endomembrane system is a collection of membrane-based organelles that work together to create, modify, and export cell products such as proteins and lipids. It consists of the nuclear envelope, the rough and smooth endoplasmic reticulum, the Golgi apparatus, and several types of vesicles. Let's start our tour with the endoplasmic reticulum. There are two types of endoplasmic reticulum, rough and smooth. The rough endoplasmic reticulum, or rough ER, is studded with ribosomes. These ribosomes are what give the rough ER its rough appearance. Ribosomes are responsible for producing proteins, which are packaged into transport vesicles and carried to the Golgi apparatus. The smooth endoplasmic reticulum, or smooth ER, lacks ribosomes on its surface. This is why it's called smooth. The smooth ER is mainly responsible for the detoxification of harmful chemicals. It's also responsible for the production of lipids, which are also packaged into transport vesicles and sent to the Golgi apparatus. The Golgi apparatus is made of a series of flattened sacs that look a little like pancakes. Its job is to package and distribute cell products that are either destined for a specific location within the cell, such as the plasma membrane, or for secretion from the cell. You can think of the Golgi apparatus as the post office of the cell, where packages are dropped off and destinations are assigned. For example, some of the proteins from the rough ER are modified by the Golgi apparatus to become digestive enzymes, which are packaged into a special type of vesicle called a lysosome. The job of this particular lysosome is to fuse with another vesicle coming in from the plasma membrane. The lysosome's digestive enzymes then break down the contents of the vesicle, releasing nutrients for the cell. Other cell materials leaving the Golgi apparatus may be destined for secretion from the cell. For example, some might be hormones that help with body regulation, while others might be proteins that become part of the materials surrounding the cell. Before these products can be exported from the cell, they must first be packaged into a secretory vesicle. This vesicle then merges with the plasma membrane, releasing its contents into the external environment. The endomembrane system can be modified slightly to meet the specific needs of each cell. For example, a muscle cell usually has more rough ER because of its need for protein, while a liver cell usually contains more smooth ER because of its role in detoxification. Regardless of these modifications, the movement of materials within the endomembrane system is basically the same for all eukaryotic cells.
So I hope that you found that tour through the cell and the video on the endomembrane system useful in preparation and for the seminar task. And for those of you not doing the seminar task, it's additional information for you to use. So until next time, this is the Opinionated Science Educator, and I will see you next week.